Good morning, everyone. So here are my disclosure statements. You guys can read them faster than I can. Let's see. Something. There we go. So as you heard already from Peter and the first two speakers, the whole genome sequencing tools are already game changers in terms of outbreak detection. We detect more and smaller outbreaks. We can detect new pathogens more rapidly. We can better characterize pathogens. But what I really want to focus in and build on what Peter said is that whole genome sequencing and other genomics tools are not magic bullets. There's continued, and exactly what Peter said, and maybe increased need for good epidemiology to identify outbreak sources and to use those data. And there's also a tremendous need to understand the complex supply chains we have in the industry as we interpret those data. So the mysteries that remain with whole genome sequencing are very much on the interpretation side at the end of it. Importantly, what we need to keep in mind is that these tools cannot and will not rewrite biological paradigms, and I'm going to show you what I mean with that. So, mystery number one, what does a SNP difference mean? Peter said that there will not be a magical cutoff. 5 SNPs and less is closely related, it's an outbreak, 6 and more is not. Will not happen. It will always be a mystery to some of us what that interpretation of a SNP cutoff is. And that's a reflection of the biological paradigm. Bacteria divide into 2 identical copies, mutations accumulate. The speed of the accumulate depends on generation times. If they grow faster, mutations happen faster. If they sit around in a dry food processing environment, they might not accumulate the mutation, it might not become different over years, maybe even decades. WGS data only tell us that 2 isolates share a recent common ancestor. They don't give us cause and effect, they don't give us directionality. Therefore, epidemiological data are essential, but they're not always going to be easy to ascertain. And that's what I'm going to really, on my mysteries number 2 and 3. Mystery number 2 is, how do we acquire and interpret epidata in a world that solely relies on whole genome sequencing? I'm going to propose that the traditional epi methods, case control studies, will not work for the small clusters, the small outbreaks we detect, because we can't run the same stats we could when we had outbreaks with 70 and 100 cases. And it will particularly not work if we have the infamous outbreaks of 1 or 2. Case control study data, as we detect more outbreaks, are difficult to acquire, they're expensive, and therefore we're starting to use historical food consumption data. Here's an example from a recent outbreak report where we looked at a food consumption history on a 2018 outbreak and compared it to 2006-2007 data, which is becoming more and more the standard in the US. Now, we will get new data every 10 years, but food consumption patterns change in people. So how do we run epidemiology? How do we use that? Because we need epidemiology because there isn't going to be a magic cutoff for SNP differences or allelic differences. And then we run into other problems. We now have outbreaks or recalls announced, and then epi investigations sometimes will start later because we have a recall, we whole genome sequence the isolate, we find some matches, then we start the investigation. Now people already have in their mind certain products that are recalled. We will see recall bias. Number 3, and this now moves more to the interpretation of whole genome sequence data by FDA, USDA, and agencies that are really sort of involved more in the monitoring of the food supply chain. What does it mean if we find a similar isolate, the same isolate, with some SNP differences in a facility 2 or 3 times? Does the organism persist there? Does that mean that under unhygienic conditions, food produced under unhygienic conditions is adulterated? It needs to be recalled. But how do we know it persists there? How do we know it's not being reintroduced? Do we have enough data to make those decisions? How do we make those decisions? That does rely on a lot more than whole genome sequencing. So the mysteries with whole genome sequencing is SNPs and interpretation, but the real mysteries are how we use these data in the real world and interpret them correctly. So biology is going to remain mysterious. We need to deal with those mysteries. Thank you.